I'm going to be telling you about my project trying to understand predisposition to esophageal adenocarcinoma by looking at defects in genes associated with DNA damage repair. And I am well aware that I am the person in between us and lunch, so I'm going to try to stick to my time slot. But before I jump into the project, I thought I'd tell you a bit about me. My undergraduate degree was in mechanical engineering and arts. So once upon a time, I thought I would have a totally different career path. But I very quickly realized that that was not the career for me. I was very lucky to have done a lot of science in my undergraduate degree, and I landed a casual research assistant role in the human genetics lab at QIMR Berghofer. And it really gave me my first taste of cancer research. I spent a lot of time processing bloods and extracting DNA and getting things ready to be sequenced. I then moved on to the drug discovery lab, which was, it's an amazing lab. They collect kind of bits of plants from the rainforest, sort of leaves and fruits and berries, and they break them down into different compounds to try to identify new anti-cancer drugs and anti-inflammatory drugs. And so while I was there, I was taking part in creating these compound libraries that could be used to screen against different cancer cell lines. I was pretty sure at this point that I wanted a career in cancer research. So I started my PhD looking at familial melanoma. And so that's really given me the skills, I think, for this project that I'm going to be talking about today. So during my PhD, I spent a lot of time looking at the DNA from different families, trying to understand the genes that might have caused increased cancer risk. And I was really fortunate to be working with not only some big groups around Australia, but also some big international research groups as well. And so when I finished my PhD, I was very fortunate to be awarded an NHMRC Early Career Fellowship. And I came here to the Surgical Oncology Group at the UQ Fraser Institute. And our real focus here is on cancer genomics. So I predominantly work on esophageal adenocarcinoma, but as a group, we also work on pancreatic cancer and melanoma. And we really have a very clinical focus, trying to look for positive outcomes for patients and looking at ways that genomics can be integrated in, into the clinic. So there are two types of esophageal cancer. There is esophageal squamous cell carcinoma, or SCC, and esophageal adenocarcinoma, or OAC. So my project is focused on OAC, and I'll use that acronym kind of moving forward. There are quite a few risk factors for esophageal cancers, some of which are related to diet, such as obesity and then smoking as well. Reflux is also a big risk factor. And then there is a fourth condition, which is called Barrett's esophagus. And this is where the lining of the esophagus changes as a result of being subjected to reflux for long periods of time. And when patients develop Barrett's esophagus, they have an 11 times greater risk of developing OAC. So in Australia, the incidence of OAC has more than doubled over the past 30 years, and the rates are still climbing. The overall prognosis for these patients is quite poor. Even when they're treated with curative intent, greater than 50% of these patients still have recurrent disease. And at the moment, all relapses are currently incurable. So it's really important that we can advance our understanding of how these cancers develop, so we can enable earlier detection we can improve the treatment options and ultimately increase the chance of more successful treatments for these patients. So currently, patients are offered a combination of chemotherapy and radiotherapy followed by surgery, but only about 40 to 50% of tumours actually respond to this treatment. Very recent trials have shown that patients that have later stage disease can benefit from being offered immunotherapy. But even so, many patients have no benefit but can have irreversible side effects. And really what we're seeing is that immunotherapies are working, I think, are more effective in patients with SCC compared to OAC. So understanding which patients respond to the current therapies, including immunotherapy, is still a really big clinical challenge. And there are probably many reasons for this, but one of the big ones is that esophageal adenocarcinoma is very heterogeneous. So when you look at tumours between patients, no two tumours look the same. And even when you look within one patient, the tumours can be quite different. So getting a better understanding of the genetics and the genomics driving this will hopefully enable a more precision medicine approach. So what we know from the literature is that up to 10% of cancers are caused by inherited or germline gene variants that give a person an increased disease risk. One of the most common examples of this that people have often heard of is BRCA1 and BRCA2 in relation to breast cancer. These types of variants are very rare, but when they do occur, they do confer a very high risk of cancer development. And what we see overall is that many of these types of variants occur in DNA damage repair genes, or DDR genes. So this is the focus of my project. There is very little in the literature looking at esophageal adenocarcinoma and germline risk. There are a handful of studies that make some association. The first gene is P53. Somatically, this is mutated in about 80% of these tumours. But when it's mutated in the germline, it gives rise to a condition called Li-Fraumeni syndrome. And esophageal adenocarcinoma is part of that cancer spectrum. There are also reports of ATM being involved, which is a breast cancer gene, and also BRCA1 and BRCA2. There are quite a few hypotheses that are associated to this study. The first is that DNA damage repair pathways are important in esophageal adenocarcinoma. Because the esophagus is constantly exposed to damaging factors that are related to reflux and to diet. We hypothesize that there are high impact germline variants that would contribute to susceptibility to this disease, that these variants could impact clinical outcomes for patients, including the incidence of Barrett's esophagus, the overall patient survival, and the age of onset of disease. We hypothesize that these gene variants could identify Barrett's esophagus patients that are most at risk of developing cancer. And then finally, that DDR genes can be used to identify a subset of patients that may be sensitive to DNA damage repair targeted therapies, such as PARP inhibitors. So the aims of the study are to first identify high-risk germline mutations in DNA damage repair pathways, to secondly determine whether these pathways are overrepresented in cancer patients compared to patients with Barrett's esophagus only, And then thirdly, to determine whether secondary somatic events in this pathway could contribute to overall cancer development. So this is a study workflow. At the top, it starts by creating a data set. Hopefully that will reach very close to 1,000 patients. So we're very fortunate in our lab to run a biobank that that recruits patients to our study and allows us to access a range of samples. So we are sequencing 360 patients that have been recruited through our biobank. We're also accessing publicly available data through the Cancer Genome Atlas, as well as data sets that have been published in the literature as well. And so we're looking in this first step at the germline information. We are then going to identify variants that occur in DNA damage repair genes or other genes that we know are associated with cancer risk. And to determine which variants are pathogenic or likely pathogenic, we're going to access public archives such as ClinVar, which contain evidence for, I guess, the risk associated to different variants. We will then sequence the tumour from these patients or access that tumour information to identify second hits occurring somatically. The third phase will be to look at clinical associations. So this will include the age of onset of disease, Barrett's esophagus status, what the patient's treatment was, and what their survival looked like. And so overall, this project is aimed at improving health outcomes by identifying these DDR defects and determining whether they're overrepresented, understanding whether they confer a higher risk of developing esophageal adenocarcinoma, and trying to understand how this impacts patients. And so I have some preliminary data from the first 300 patients. We're still in the process of aligning the rest of the data sets to the current human genome. So still a little ways away from that full 1,000 patient set. But what we have seen is that out of the 300 patients, we were able to identify 41 pathogenic or likely pathogenic variants in 38 patients. These variants occurred most frequently in ATM and in BRCA2, but also occurred in a range of other genes associated to DNA damage repair. We found that 50% of the variants were in the homologous recombination pathway. And an interesting finding was that we identified 3 variants occurring in something called the mRN complex. So this complex is involved in the processing of double-strand DNA breaks, and it binds the double-strand breaks to tether the ends before it can be repaired. So we found 3 individual patients that had a pathogenic variant occurring in each of these genes. This complex hasn't been associated to esophageal adenocarcinoma previously, and this is still very early evidence that perhaps it could be playing a role. So this is something that I'm looking to investigate further. Interestingly, this complex activates ATM in response to DNA damage, and ATM by phosphorylating check 2 is also involved in recruiting the BRCA complex. So overall, I think the picture this is giving is that we're seeing a host of interrelated genes that could potentially be important in this germline risk factor. I've also tried to look at some of the clinical features, though with this smaller data set, it's still a bit early days to draw big conclusions. Firstly, what we saw is that patients with Barrett's esophagus actually had improved survival compared to patients that did not have Barrett's esophagus. And then when we looked at the age of onset of disease, though it is not significant, we're seeing a trend that patients that have one of these DNA damage repair variants are actually presenting with disease at an earlier age. So it'll be really interesting to see how that looks once we're able to assess the full cohort of patients. And then this leads me to the long-term goals of the project, which is that we will be able to identify these high-risk variants and use them to tailor treatment on a personalized basis. And this research has not yet successfully been conducted in esophageal adenocarcinoma. Currently, in other cancer types, patients that have defects in these genes can be treated with platinum agents as well as PARP inhibitors, but PARP inhibitors are not yet standard of care for esophageal adenocarcinoma. So the big picture is that integrating genetics into clinical practice could be a positive shift in the way we treat these OAC patients. Big thank you to my research group led by Professor Andrew Barber and the clinicians at the PA hospital that help us with our biobanking program. Thank you to the bioinformatics team at QIMR Berghofer and to the patients and their families that make this possible. And a big thank you to Cure Cancer that supports this project. Thank you.